So for the experiment this week, we are trying to investigate how well the DNA sequence S33, S34, and S35 expressed by investigating the production of pale A for the bacteria E. coli. And we have 12 cultural dishes for each of the DNA sequence, so 66 in all, and for 6 of each we put it sealed with the parafilm to leave it anaerobic, and other 6 we just do nothing for it to leave it aerobic. And for each day, after the first day, we will scrape 2 culture dishes and seal them under -20 degrees. And on the first day or the Thursday, we will see the OD for each of the bacteria. So I will talk about the procedure briefly. So how we scrape off the pale A bacteria. Basically, we first add 2 milliliters of M9 solution into the culture dish with those bacteria. Then we scrape it off and put them into a tube. And later, this is 2 milliliters, and later we'll do it again to resuspend them and make sure all of the bacteria were in the tube. And at last, we'll put them into 5 milliliters by adding more M9, which is a 5 milliliter system. And so how this experiment goes. An unfortunate scene is that on the third day, we are doing this like live growing for 24 hours, 48 hours, and 72 hours, and to see like how it grows in the independent variable are first, of course, time, and also we are seeing the effect of oxygen or anaerobic and aerobic situation. So on the third day, we examine the OD of the solution, and I will also talk about like the way of examining, like its mechanism. Basically, we have our bacterium here, and it will grow PA, and there's some his tag attached to it. And at first, we'll put the first antibody onto it, which will recognize the his tag on that. And later, we will then put the second antibody, which will attach on the first antibody, and also it has a HRP enzyme attached to it. So then when we add the color expressing agent or color presenting agent, you will turn the solution into brown if there is pale A. So basically, the more brownish it is, the more pale A it has. But unfortunately, our OD is really low. Basically, it's because on the second day we already found that, this is some of the bacterium growing on it, and we found some hollow points on it that no bacteria exist. So we think that there is some bacteriophage on it that eats all of us bacteria, so it's basically growing thinner and thinner with less protein as the time increases, which seems to be not plausible unless there's bacteriophage. So in this case, we know that our lab is contaminated with the bacteriophage, so we have to conduct this experiment again. So that's what we are doing basically, and this week is kind of like a repetitive job. Like we know we have to do 36 dish